Okay. In Chapter 2, we cover two processes that you've almost certainly learned a couple of times throughout your educational career, and those are mitosis and meiosis. And you may think that this is just going to be a rehash of much of what you've already learned and will be relatively straightforward. And for the most part, that's true. There are, of course, some finer details that we're going to dig into, but there are some really important things that you need to understand that differentiate mitosis and meiosis from one another that come into play later when we talk about DNA replication, when we talk about mutation and heritability. All of these things are really important to understand. And if you don't have a solid grasp of mitosis and meiosis and the differences between them and how they relate to one another, which is they don't really, you're going to have a harder time understanding that. So, without further ado, let's jump in. The first thing we're going to do is dig into some background material, some foundational knowledge that you've got to have. And the first is, what's the difference between these two? How is genetic material passed from parent to offspring or cell to cell? And those are differentiated between mitotic and meiotic processes. So the passing of genetic material from one cell to another, okay, when a cell divides into two, it ensures that both of those descendant cells have the exact same genetic material. Okay, that's important because every time your cells divide, you don't want them to end up with half of the genetic material they had to begin with. This process is mitosis. So much of this is allocating cellular resources to the two descendant cells, but we've also got to ensure that the genetic material is faithfully copied and passed along to each of the descendant cells. Now, when information is passed from parent to offspring, that is part of the meiotic process, or meiosis, the process whereby we as humans produce gametes and other organisms may produce things like spores. Okay? So we're going to dig a bit into the structure and function of eukaryotic cells here. A lot of this is rehashed. These are just some of the organelles that are important for our discussion of mitosis and meiosis and subsequent discussions of genetics. Okay? So the first, of course, is the nucleus. This membrane-bound organelle is where all of this genetic material resides. And this nucleus is illustrated, and the genetic material in this nucleus exists in multiple states at once for the purposes of illustration. It doesn't generally exist this way, but for now it does, okay, for this illustration. The first term I want you to become familiar with is chromatin. And you can see that the word chromatin is pointing to just this amorphous, gooey looking area in the nucleus. And this term describes the general state of our DNA while our cell is undergoing normal operations. While it's doing its thing, generally the DNA exists in this state of chromatin. Now, you will quite frequently hear us talk about chromosomes, and when you think of your DNA, you probably think about chromosomes, okay? Chromosomes, illustrated here, are DNA that has been highly compacted and condensed into a very easily movable form, okay? If it is in that very loose, relaxed state of chromatin, it is not as easy to move about, and as you will see when we get into mitosis and meiosis, it's critical at these times that the chromosome is easy to move, okay? And then we also have the nucleolus. We won't discuss this much more in this course, but just know that this is where RNA is made, which happens during the process of transcription, and then also where ribosomes are assembled, okay? Now, one thing I want to point out in this image is the structure of these two, well, there are 3 chromosomes, 2 of them are pointed to. You'll notice that they look kind of like an X, and I'm going to refer to them as X's for simplicity's sake throughout this course. Now, what I want you to recognize, and should become very clear here shortly, is that chromosomes are often illustrated. Whenever you do a Google search for chromosome or genetic material or DNA, you'll see this image of these X's, okay? What this X represents is something very different from the 2 chromosomes you saw on the karyotype in chapter 1. Remember, you saw 2 chromosome number 1 side by side and 2 chromosome 2s side by side. But if you will recall, I told you that those were an image in which someone digitally cut them out and lined them up side by side. You'll also notice, if you go back and look at those, that they are not physically touching one another. Whereas in these X's, each half of the X, each side, is kind of tied together in what you might call, looks like a waist. Okay, this is actually the centromere. We'll get to that in a moment. But the point is, when you see a chromosome that looks like this, is in this X shape, it represents something very specific, and it confuses many, even illustrators, okay? What this is illustrating to you is a chromosome that has already been replicated. It's already been copied in preparation for mitosis or meiosis. If your cells right now are doing their thing, you do not have these X's. You have this relaxed chromatin state. And even if you were to take all the chromatin in that cell and condense it, you wouldn't have this X. You would have just 1/2 of that X, because your DNA has not been replicated unless you're preparing to enter mitosis or meiosis. So we'll dig into that in a bit, but whenever you see a chromosome illustrated, I want you to think very critically about whether it's being illustrated properly, and your understanding that you derive from this chapter should enable you to do that. So prokaryotes, unlike eukaryotes, they use DNA, but they don't have these membrane-bound organelles that we have, things like a nucleus or mitochondria, okay? And while their DNA does get compacted, it's not supercoiled into these really tightly bound chromosomes like eukaryotes have. Now on to the other organelles of the cell. I'm going to briefly go over these 4. And the first of these is the endoplasmic reticulum. Okay, you can see here that the endoplasmic reticulum, in this illustration, it's a membrane branching off of the nucleus, and there are 2 portions of the endoplasmic reticulum. Towards the top side, you can see that that region is classified as the smooth ER, and that's where fatty acids and phospholipids are synthesized. And that'll be the last time we mention the smooth ER in this class. The rough ER, on the other hand, is an incredibly important portion of the cell and an important organelle, and it is where ribosomes translate RNA into proteins. Okay? You will remember from the previous slide, in the nucleolus, the RNA is made, as are ribosomes. Those things come out of the nucleus, and they work through the rough ER in this process that we will learn about later called translation. Okay, so the endoplasmic reticulum, it's involved with the ribosomes in basically creating all the proteins and things that your cells make. All right. Now, the other organelles that I wanted to touch on, mitochondria. These are found in plant and animal cells. You probably learned of these as the powerhouse of the cell, or the energy source of the cell in early biology classes. These are the sites of oxidative respiration. They generate ATP, right? And so because this is where ATP is generated, that's why they're called the powerhouse of the cell. Okay, we're going to continue talking about mitochondria here in a moment. Chloroplasts are quite similar, although they're not found in animals. They are found in plants, including algae and protozoans. They produce energy through photosynthesis. Okay, so this is also something that you've learned, but something that you might not be aware of is that both chloroplasts and mitochondria have their own DNA. Okay, so apart from the DNA that you have that is in your nucleus, all those mitochondria floating around in your cells, those mitochondria are outside the nucleus in the cytoplasm. Each of those mitochondria has its own circular chromosome, okay? And on that chromosome, there are genes, just like the genes that you have in your DNA in your nucleus. Mitochondria and the DNA inside them is duplicated. They go through the same processes of transcription and translation that you do within your nucleus. And most of the time, these organelles are maternally inherited. Okay? And what that means is when sperm and egg came together and created you, the sperm carried nothing but chromosomes. Okay? What the egg carried is lots of cytoplasm. It carried with it mitochondria and other organelles that you've probably learned before. But all of this extra stuff comes with the egg. Think about a chicken egg versus chicken sperm, right? One makes a hearty breakfast, the other you can't even see, okay? And in that egg is a great deal of material, including these mitochondria, and that is why they're maternally inherited. And if you zoom in and look at, this is a schematic of a mitochondrial chromosome, you can see that there are all these genes on here. 16S rRNA, 12S rRNA, cytochrome B, NADH dehydrogenase subunits. So we'll dig into kind of what these are later in the class, but each of these highlighted blocks represents a specific gene in the DNA of a mitochondrium that is transcribed and translated and proteins are made, which is remarkable. So why do we have these little organelles within us that have their own DNA? Well, it is clearly the result of the process hypothesized and given the name the endosymbiotic hypothesis. And so this is the idea that mitochondria and chloroplasts, what they really are, they used to be endosymbionts, other living organisms, bacteria, that lived and were associated with cells, living plant and animal cells, and then became assimilated and became obligate endosymbionts, such that the organisms, like you, can't live without those little bacterial cells within their cells. So basically, you can think of each of your cells having a bunch of little bacteria inside them that are doing work for the cells. Those mitochondria are doing work for your body. Your cells would not survive without them, okay? If we look at a phylogenetic tree illustrating the relationships of living organisms, you'll see we are up at the top along with other eukaryotes. So there's animals, our closest relatives are fungi and plants and ciliates and slime molds. There's another giant group of organisms or clade of organisms, the archaeans, and then at the bottom you see bacteria. If we look at the DNA of our mitochondria and our chloroplasts, and we were to analyze it and compare it to all living things, the DNA of our mitochondria is most similar to purple bacteria, okay? It's very similar, has very similar genes. Some of those genes are in the same order. And this tells us that mitochondria are basically, or they used to be, a purple bacteria that was associated with early eukaryotes and became assimilated and obligate partners with our cells. Now, when this was first hypothesized, the woman that proposed this idea was largely ridiculed, but she was absolutely proven correct. And despite the fact that this seems to be a remarkably outlandish, wild idea, it's been substantiated by recently observed instances of obligate intracellular associations of algae and bacteria. So here's a great example. What this person's holding in their hand is an egg mass from a spotted salamander, okay? So you can actually find these in ponds in northern Mississippi in early winter. These salamanders go in and lay their eggs, and the egg mass swells. It's this really dense jelly, so you can pick it up as this person has. And inside you will see their individual little eggs, look like kind of green circles with a dark line down them. That dark line is the forming salamander embryo. But you'll notice that these circles are green, like each of those eggs has a distinct greenish hue. And what's amazing is it was thought that there's algae growing on these eggs, and the algae, because they're laying eggs in these ponds that have low water movement, right? There's not a lot of aeration in ponds, right? Imagine if you have an aquarium with a fish that needs to breathe air, you've got to have an air pump that pumps air into that aquarium, right? Well, these salamanders are the same way. You drop them into water and there's no air in the water, like a stagnant pond, they can't respire. But it was thought that they have algae on the surface of these eggs, and these algae are undergoing photosynthesis. And you know that photosynthesis, you take in CO2 and you produce oxygen. So this algae is producing oxygen that helps these eggs survive. That was the idea. Turns out, it was recently discovered that what's really going on here is that these algae actually live inside the cells of the female salamander, and she passes them on in the eggs that are fertilized by males. And so these larvae have algae within their own cells that are producing oxygen and help them survive in these low oxygen environments. Pretty remarkable. Okay, on to a few other cellular structures that I want you to be familiar with. The first is centrioles. Okay, now centrioles are these microtubules that exist in the cell at all times, but they play a critical role in mitosis and meiosis. Okay, and we'll come to that role here shortly, but just understand that centrioles are associated with spindle fibers, and they're used primarily in metaphase and anaphase of mitosis and meiosis. And then the final term or structure that we're going to talk about, and I want you to understand, are spindle fibers. So these are microtubule fibers that attach to the chromosomes, the replicated chromosomes. You'll see what we've got here is those dark green stripes that go side to side. Those represent a replicated chromosome. Remember, I've talked to you about when they're bound together like an X. We're looking at an X on its side. That replicated chromosome has a spindle fiber attaching to the bottom portion of that centromere and another spindle fiber attaching to the top portion there. Remember, I told you that constriction where they're joined together is called the centromere. It's kind of like the waist is the analogy I use. And on either side of that centromere, you have what are called sister chromatids. Okay, so that X there, that dark green replicated chromosome, that's a single chromosome, and each half of that X is identical to the other. We'll get way into that, okay? But I want you to understand that each of those halves is a sister chromatid, as you see here illustrated, not a chromosome. There's only one chromosome in that image, okay? And on the right, there's a fluorescent image of a bunch of chromosomes in blue with their centromeres in pink and many, many, many spindle fibers that are connected to each of those centromeres, okay? Now you'll notice that all of those bright green spindle fibers appear to emanate from a dense point at the top and bottom of that image, okay? And you can think of that as an anchor point from which these spindle fibers will attach themselves and pull apart these chromosomes. That's the fate of this chromosome here. Each of those two sister chromatids is going to be ripped apart from one another. That centromere is going to get split in half, okay? That tearing and pulling is accomplished by the spindle fibers, and it is because they are attached to the centrioles, okay, from the previous slide. So we'll dig into this. All right, now, I spoke to you in the previous lecture in chapter 1 about homology. Remember, each chromosome that you have two copies of chromosome 1. They are homologous to one another. One you got from mom, one you got from dad. So because you got them from different parents, they are not identical. Certainly your parents have different alleles, different genetic backgrounds that they've inherited and passed to you. So those two copies of chromosome number 1 are homologous. Now when we get into mitosis and meiosis, those chromosomes replicate, each of them. So each of your chromosome 1s that you got, the one from mom and the one from dad, is going to replicate and turn into this X-shaped chromosome. So you'll have two X's that represent your two copies of chromosome 1. Each of those X's is homologous to one another. Each of the sister chromatids within those chromosomes is identical to the other. Okay, so each half of an X is identical to the other half of the X. Okay. Now, when we look at the structure of chromosomes, here's some schematic illustration of replicated chromosomes. None of these are homologous to one another. The one on the far left is obviously not the same chromosome as the one to the right of it. And those other two to the far right are much smaller. They're oriented very differently. All of these have their centromeres in a different relative position. And so this tells us that these are very different chromosomes. This could be chromosome 1, chromosome 2, chromosome 18, and chromosome 19. Okay? Now, each of these has been replicated. You know that. It's been copied because it exists in this X form where there is this centromere, this primary constriction, and above and below this centromere are the arms of the chromosome. There's an upper arm and a lower arm. And that placement of the word centromere on the top left is not pointing at the right spot, obviously. It should be right in that area of primary constriction. I just show you this so that you can realize that the centromere is not always in the exact middle of a chromosome. For these acrocentric and telocentric chromosomes, they have their centromeres at very different positions. Okay? You don't need to learn these terms, metacentric, submetacentric, acrocentric, telocentric. Don't worry about that. I just want you, when you see a replicated chromosome, quote unquote X, one of these X's, I want you to know what it is, that it's a replicated chromosome, not two homologous chromosomes. And I want you to realize that the centromere of these chromosomes means something, and it tells you about whether or not you're looking at whether these are two homologous pairs of chromosomes or these are four different chromosomes. All right. Now you've already seen a karyotype. This is where a karyotype comes from. It's a photograph taken of a cell when the genetic material has gone from that relaxed state of chromatin that I described to you already, and it condenses into these solid chromosomes. That's how we get these images. Like this human karyotype for a male, that, like I described in the previous slide. So here we have 23 pairs of chromosomes. Okay, so total you have 46. How many did you get from mom? 23. How many did you get from dad? 23. Look down at those sex-determining chromosomes. Let's imagine this is my karyotype. Okay, I have a Y, that's what makes me a male, and I have an X, that's what helps me make some rational decisions. And of those two, the X and the Y, I got one from one parent and one from the other. I bet you can guess which parent I got my Y chromosome from, okay? And which one I got the X chromosome from. Now look at chromosome 22. Can you tell me the same thing? You can't. Just by looking at the two chromosome 22s that I have right there, all you know is that those are both 22s. They have the same genes, but I got one from my mom, one from my dad, and they may be identical at some genes and they may be different at other genes, okay? But every individual, unless you have a genetic mutation, is going to have 23 pairs of chromosomes and therefore 46 total chromosomes. Now from this, I want you to understand that we are going to use this letter N to signify the number of chromosomes, the base number. So for humans, that number is 23, okay? We have 23 distinct chromosomes. And if I gave you all N of my N human chromosomes, I gave you a chromosome 1, a chromosome 2, a chromosome 3, all the way down to 23, you would have one copy of every gene. But that's not what a living individual has. Each of us has 2 copies of every gene, 2 copies of every chromosome, except the sex chromosome. Only females have 2 copies of the same thing. And the haploid number, N, is the basis. That's a full set of chromosomes. So you have 2 full sets, I have 2 full sets, okay? And if we look at other organisms, genome size is highly variable, okay? So some things like African wild dogs have 78 chromosomes. So badgers have 32, carp have 104, adder's tongue, the small plant, has 1,200 chromosomes. and cotton has 52. Now looking at these, what did I tell you the 2n number is for humans? What's 2n for humans? It's 46, okay? So what is n for humans? If I tell you 2n, if I give you a math problem, I know this is genetics, but you're going to do some math. 2n equals 46. Can you solve for n? Surely, it's 23. So if 2n for a carp is 104, what is n for a carp? 52, okay? So you can, if I tell you an organism is 2n, you can tell me what n is. You can tell me how many chromosomes are going to be in a sperm or an egg for that organism, okay? All right, so now we're ready to move on to the cell cycle, which is an important part of mitosis, okay? This basically, if you look, most of the cell cycle is not mitosis, but we spend a ton of time talking about mitosis, just this small little bit of the cell cycle. It's because the rest of it, interphase, you see how interphase is everything that's not mitosis, is the basic life of a cell, where it grows and doesn't do a whole lot more. It's not dividing. It may be preparing to divide, but it's not dividing. So mitosis is when the cell is dividing into two. Everything else is interphase, okay? And we're going to go through all these phases and discuss the characteristics that define them, okay? So the cell cycle, when I say the cell cycle, what I'm talking about is from the beginning of the existence of a cell, and that existence, unless it's a zygote, every cell in your body that is alive right now, every living cell you have, originated from the process of mitosis. And so it had a birth date, and from that point to the point when it begins to divide and go through mitosis itself, that's the cell cycle, okay? So that is all interphase. And there are three parts of interphase, G1, S, and G2. The G's refer to gaps. There are things going on, but not what happens in S. S, the big thing in S, is we replicate DNA. So you know what, if I've got a cell, if I've got a nerve cell in my brain, it's never gonna divide. It just came into being early in my development, and it is existing as a nerve cell in my brain. It is not going to go through S, it is not going to go through G2, it's not going to divide. It just exists and does its thing. That's part of the cell cycle for it. Now other cells, like a skin cell on my arm, when I get a cut, they existed in G1 until I got that cut. At that point, they're like, holy crap, we've got to fill in this gap in this skin. And so they began the process of going through the S phase in G2, and then mitosis. So I get more skin cells to fill in that gap, okay? Now in interphase, as I've mentioned, the DNA exists in this relaxed state of chromatin, okay? So it looks here like a bowl of ramen in the middle of this cell, in the nucleus. It's a very simple state. We don't need to talk a whole lot about what's going on in interphase, but I want you to understand that the DNA does not exist in this tightly bound chromosomal form. There's no X's, there's no bars like you see in the karyotype. During interphase, it all looks like this. Okay, so let's keep going through the cell cycle. We're going to dig into this. As the cell cycle begins, the cell is basically resulting from a previous mitotic event. So right under where it says cycle begins, you see that cell's kind of tiny. And if you track around G1, the cell is growing, okay? Now, our cells are necessarily smaller than the cell that split in two, right? The cell that went through mitosis, that divided, one cell became two, so it's necessarily smaller than it was initially. And so the cell has to basically bulk back up. It's got to grow and be prepared to divide itself. Otherwise, from the time you're conceived to the time you expire, your cells would decrease in size every single time they divide, okay? And your cells would be incredibly small. All right, so what we're working towards is this process of the S phase where DNA is replicated, and then G2 where the cell prepares for division or mitosis. Now, I've already mentioned chromatin. It's this relaxed state of DNA, okay? The chromosomes that you see later, they are an incredibly high compaction of this relaxed DNA. Your DNA, when you can see it under a microscope as a chromosome, is compacted 5,000 times, okay? So you've all seen a car that's put in a car crusher and smashed into a cube. Imagine taking an automobile, like a big Chevy Tahoe, and compacting it 5,000 times so that you end up with a piece of metal that is 1/5,000th the size of the full-size Tahoe. That's the amount of compaction that we're talking about when we go from chromatin to chromosome. Now, during normal cellular function, our DNA exists in this relaxed chromatin state because it's most easily translated, it's most easily accessed and read. Okay? The DNA needs to be open and loose and accessible for our cell to be able to go to genes on our DNA and read them and make the proteins that are coded for there. Okay? It's just like your textbook. Okay? It's a lot of information very densely packed into very thin sheets of paper. If you don't open it and spread those sheets of paper out, you can't access that information. Okay? Now, what we see happening in G2, so this is after the S phase. The only thing you need to know that happens in the S phase is we double the amount of DNA. Okay? DNA is replicated. So we've got twice as much ramen in this bowl than we did before. Okay? In G2, we've got all of this DNA in the cell, and it's going to start condensing. Okay? We have intracellular mechanisms that are going to make sure when we replicated that DNA, didn't make any mistakes, right? Because we are basically copying 3 billion nucleotides, G's, A's, T's, and C's, making copies of them pretty quickly, okay? And in order to ensure that we have faithful copies, they need to be checked, okay? If we don't check it, if we're not proofreading that replication, that's how we end up with genetic mutations, some of which lead to serious diseases like cancer. Okay, so proofreading, checking that 3 billion nucleotide photocopy, if you will, of DNA during G2 is a critical step. Okay. All right, so now we're ready to move on to mitosis. And this is when chromosomes divide and become 2 daughter cells. So we've got a very simplified set here where 2n equals 2. Okay, n is 1. There's a red one from dad and a blue one from mom. Okay, we've got 2 chromosomes in this cell. The DNA replicates. Now you see we have 2 X's, 2 replicated chromosomes. And then we go through mitosis, we'll dig into that. And at the end, we end up with 2 cells, 2 diploid cells, diploid meaning 2n, that are identical to that first cell that we started with. As you can see, they have the exact same amount of genetic material. All right? And so this is the process that we're working towards through mitosis. And I'm going to detail through discussion of these phases how in mitosis this is accomplished. Okay. So the first thing we need to recognize is that, remember looking at the cell cycle below, we exit G2. That's where the DNA condenses now as we exit G2. The DNA has been replicated, so we have these replicated chromosomes, or the X's, all right? And we should be comfortable now with this chromosomal terminology as we move forward. I want you to be certain that these are terms that are familiar to you and you understand the difference between them. Chromatin, homologous chromosomes, and sister chromatids, okay? Remember the difference between the sister chromatids and homologous chromosomes. It's huge. It's a huge difference, and it's critical you understand that difference, okay? All right. So the first step of mitosis is prophase. And you can see here not a whole lot has changed other than that replicated DNA has condensed into chromosomes. And here they don't so much look like X's as two spaghetti noodles that are stuck together to one another, but they still have a centromere, okay? So each of these chromosomes that has been replicated consists of two sister chromatids, all right? You'll also notice here that the early mitotic spindle is forming, so these two centrioles have spread apart from one another, and they've got these yellow fibers sticking out from them. Those are going to be important later on, but it's the movement of these centrioles to opposite sides of the cell and the formation of these spindle fibers that is pretty important, okay? Now, one of the bullet points I have on the right clearly states that these microtubules establish an axis, okay? And by that I mean, well, I guess you could relate it to lines of latitude on the globe. When you think about the Earth, you might think about the equator being a line that splits the Earth right across the middle. Well, the terminus of each of these spindle fibers, you can see here, they all end kind of in the same spot, and it's going to create a very similar equatorial plane, or what we call a metaphase plate here in a minute. You'll also notice here that there are some holes starting to develop in the nucleus. You got to break that thing down, and we're on to prometaphase, okay? Now, this is an intermediate phase between prophase and metaphase, and named accordingly. The thing that you need to be aware of here is that this is when chromosomes are starting to move, okay? They're moving towards this equator that I talked about. But the really important thing here is that these spindle fibers are attaching to the centromeres. So you can see there is one labeled kinetochore here, and it's on the upper side of a centromere of one of these sister chromatids. You'll notice on the other side of that very same centromere, the sister chromatid attached to it, there is a spindle fiber also attached to the other side there. And so we have spindle fibers attached to both sides of the centromere of each of these replicated chromosomes. In metaphase, all of these chromosomes have lined up end to end with spindle fibers attached to either side, okay? So think about what is lined up on this metaphase plate. On the far left, that long chromosome that exists in this X-shaped form, each half of that X is an identical copy of, let's call it chromosome number 1, that came from mom, okay? On the far right is the homologous chromosome number 1. It's the one that came from dad, and each half is identical. What's going to happen here is each of those replicated chromosomes, each of those X's is going to be ripped in half in anaphase, the next phase, right here. So on the far left, we see that what was two sister chromatids attached to the centromere, identical to one another, have now been ripped apart by these spindle fibers and are being pulled in opposite directions, okay? Now, I think you can see what's going to happen here is each half, the top and bottom half of this cell, are going to become two different cells at the end of this process. And each of those halves is going to have the exact same genetic material, because each of those halves of those replicated chromosomes, each of those daughter chromosomes is identical to the other. So what's going up is identical to what's going down. Now, the final phase of mitosis is telophase and cytokinesis. So this is the step where you can see that these sister chromatids have been pulled apart into daughter chromosomes, and now they reside in different cells. We've got new nuclei forming in each one of these cells. We have a clear membrane being established that separates these two cells. The cytoplasm is being separated. Some mitochondria are on one side, some are on the other, okay? The nuclear envelope reforms and the spindle fibers disappear. And boom, we're done with mitosis. We have two cells, okay? Now, these are the images from your text, and they track the very same thing. There are similar explanations beneath the schematics with what I think are very nice microscopic images of the same process above. And so I encourage you to go through these and make sure all of this makes sense and jives with what I've just explained to you. Okay? And so you can see here at the end of telophase, we end up with, after cytokinesis, 2 different cells. Now, the only difference between plants and animals in this regard is that plants have cell walls, and so this cell wall must be rebuilt or built in between these 2 cells. But other than that, the process is indistinguishable between plants and animals. So that is the basis of mitotic division. And now I'm going to dig into meiosis. Meiosis is a process whereby the number of chromosomes is reduced by half. In our case, that number goes from 2n to n. So what that means is, instead of, like in mitosis, where we go from 1 2n cell to 2 2n cells, what happens in meiosis is that we are going to take a 2n cell and we are going to end up with cells that have n chromosomes. For humans, that's 23, not 46. Okay? So, the first awesome thing about meiosis is that the phases are named exactly the same way, and damn near the same thing happens in each of them. It's just that when we undergo meiosis, just like mitosis, DNA gets replicated first thing, okay? And just like mitosis, chromosomes line up and they get pulled apart and we split into 2 cells. The novel thing is that in meiosis, we have it happen all over again, okay? We're gonna have a whole nother round of cellular division. So what we end up with is not 2 cells like in mitosis. So we go from 1 to 2. But in meiosis, we go from 1 to 4. Now let's look at this first division, because there's one critically important thing that you need to recognize here. And if you compare these 4 phases to the 4 phases of mitosis from the previous slide, there's one thing that is incredibly important that you recognize is different here. And it's metaphase. Okay? The same stuff happens in prophase 1. This is the first meiotic division. In metaphase 1, something different happens. Okay? What we have here is a cell with 2n equals 6. N is 3. There are 3 red chromosomes. There are 3 blue chromosomes. All 3 reds came from mom. All 3 blues came from dad. Each of them has been replicated. And what happens in metaphase 1 is that the homologous chromosomes line up next to one another. Go back and look at the mitotic division. Remember that homologous chromosomes 1 were on opposite ends of this metaphase plate, this equatorial line. They weren't lined, the 2 X's weren't next to each other. Each X was by itself. What we have here is each of these replicated chromosomes is lining up next to its homologue. Okay? And when pulled apart by spindle fibers, what moves to either side of the cell and is soon to become part of 2 different cells are not the sister chromatids being ripped apart into daughter chromosomes. All that's being separated here are the homologous chromosomes that are replicated. You still have those X's connected at the centromeres. We're just moving the red one up and the blue one down, or vice versa. Okay? And this is incredibly important, okay? Because what you are going to see is that the end product is 4 cells that are not identical to one another, okay? And why do we undergo meiosis? What do we make? Is it, if I get a cut on my arm, does my cell go through meiosis? No, they undergo meiosis when they're producing gametes, producing sperm or producing eggs. Do you want all of your sperm to be identical and the eggs of your spouse, if the genders, the sex is that way, all the eggs of your spouse to be identical? Then you can only create one possible offspring. This random mixing of reds and blues on either side is where we get 99% of our genetic diversity in our offspring, okay? This random sorting of red goes up, blue goes down, or vice versa, that's it. okay? This is really, really important, and it happens in metaphase 1 and anaphase 1, okay? In meiosis 2, we have something very similar to mitosis going on. The only difference is here, in metaphase 2, you'll see what's lining up are the replicated chromosomes. In anaphase 2, they are getting pulled apart. But what's interesting is there's only one chromosome number 1 here, and it's getting pulled apart. There's only one chromosome 2. It too is getting pulled apart, and the same for chromosome 3. And so what we are ending up with is 2 daughter cells, okay, that are each going to have 1, 1, 1, 2, and 1, 3. Okay? They are each going to have 3 chromosomes. Remember, we started out with a cell that was 2n equals 6, one cell. We're going to end up with 4 cells that are n. They have 3 chromosomes. All right? So what happens in mitosis is we replicate the DNA once, and then we split the cell once. What happens in meiosis is, remember, we started by replicating the DNA, and then we split it, and then we split it again. So we doubled the DNA once and then split it twice. And because of that, we end up with cells that have half the amount of genetic material. Okay? These are gametes for humans. For other organisms, this might be how they create spores. Now, one thing I want to point out also is that you'll notice that some of these chromosomes that are mostly red or mostly blue, they've got different parts that are the other color, okay? So there's a little bit of blue on the end of one of the reds and vice versa. That illustrates a principle called crossing over, and that also happens in meiosis 1. Basically, when the 2 X's that are homologous are lined up next to each other in metaphase 1, they can get tangled up. They can get interlocked with one another, and it's kind of difficult to get them apart. And what happens is the cell ends up just cutting it and reattaching. And sometimes it gets reattached to the wrong one. But does it matter? Think about it. Let's look at, in this top metaphase 2 cell, that top chromosome that's all red on the right, mostly red on the left, and a little bit of blue on the bottom. Where did it get that blue? Let's assume that's chromosome number 2. Where to get the blue? It lined up next to chromosome number 2 in metaphase 1. Okay? And that same bit of the blue chromosome number 2 got tangled up and swapped over to this chromosome number 2. Do the 2 different chromosome 2s have different genes in different places? No. That's why they're called homologous. They have the same things in the same places. It might just be a different version of that thing. For example, an eye color gene, okay? So now it might be that that chromosome number 2 has a little bit of mom's chromosome 2 genes mixed in with dad's on that one chromosome. It's called crossing over, and it's something we will discuss in great detail when we get into discussing mutations. All right. So here are the 3 different metaphases that we've talked about. Metaphase from mitosis, metaphase 1, and metaphase 2. Look at these, look at what's lined up where on the metaphase plate, and make sure you can differentiate these from one another and understand them. And I want you to know that when I use the term metaphase, I will specify 1 or 2 if I'm talking about meiosis. If I'm talking about mitosis, I just say metaphase, okay? Metaphase 1 is in meiosis 1, metaphase 2 is in meiosis 2, and metaphase with no number is in mitosis. Always take it to the bank, okay? All right, so this process of meiosis is how we undergo the development of gametes. And for males, that process is called spermatogenesis, so the production of sperm. There are a few other terms here that I'd like you to be familiar with. These are in your text, but the undifferentiated germ cell that is diploid prior to metaphase 1 is the spermatogonium. The primary spermatocyte is what undergoes that first meiotic division. Secondary spermatocyte, unsurprisingly, undergoes meiosis 2 division. Okay, so this is spermatogenesis illustrated. Okay, and what you'll notice here is that there's really no growth of the cell after the split, and so we have size reduction, and each of these mature sperm is basically just a vehicle for delivery of sperm, and that shouldn't surprise any of you. Okay, so look, start at the top cell. Look, there are 2 blues and 2 reds. Get down to the sperm, there's 1 blue and 1 red in each. Okay, we've doubled the DNA once. That's reflected in the primary spermatocyte there, and then it's divided twice. So you end up with 1 of each, 1 blue, 1 red, in each of the sperm. Now, production of gametes in females is slightly different. It's called oogenesis, production of eggs. And what's interesting is that each of the cells that results from meiotic divisions in oogenesis gets the same amount of genetic material, but they don't get the same cytoplasm. Okay? And the oogonium and polar bodies are important terms for you to understand. The polar body is very important, as you will see in the next slide. So the polar body is an oocyte that doesn't get the cytoplasm, okay? And they don't become eggs. The process of oogenesis, unlike spermatogenesis, doesn't go from 1 cell to produce 4 eggs. That's what happens in sperm. 1 cell becomes 4 sperm. In oogenesis, 1 cell becomes 1 egg. Okay? Now there's still DNA replication. There are 2 meiotic divisions, but you'll see that these polar bodies that are the small offshoot of each meiotic division, they have the genetic material, they don't have the cytoplasm, they don't have the volume to be an egg. So only 1 egg cell is derived from the process of oogenesis. Okay? All right, so now gametes. Each gamete can have any combination of paternal or maternal chromosomes. And crossing over is an important mechanism that I want you to understand for mixing up genetic material when you produce offspring. Okay? So I'm going to put some things up that help you understand these concepts, but I've got some problems that help you understand the combination of paternal and maternal chromosomes that can arise. Okay? So if you look here in this first problem, if I ask you or tell you that a 2N cell has 3 pairs of chromosomes, okay, and they're heterozygous at each, right? So they have chromosome C. They got 1 from mom and 2 from dad. They have chromosome M. Got 1 from mom and 2 from dad. They have chromosome S. Got 1 from mom, 2 from dad. Okay? Now, what chromosome combinations are going to be in daughter cells after mitosis? And then what chromosome combinations could be present in haploid cells after meiosis? So these are 2 things that I think you need to be able to solve. So if I tell you that's what's in a cell and it goes through mitosis, what's going to be in the daughter cells, both of those cells that result from mitosis? It should be the exact same thing. Okay? You'd have C1, C2, M1, M2, S1, S2. Now, when I asked you what chromosome combinations could be present in haploid cells after meiosis, that's very different, okay? Because that cell is going to produce haploid cells that are going to get a C, an M, and an S chromosome. They'll get 1 of each. So they might get C1M1S1, C1M1S2, C1M2S1, C2M2S2, C2M1S1, C2M2S2, right? There's so many different options, okay? And that is why you can have a gazillion children and none of them will be identical to one another because each of your gametes is a random pick. Okay, we need a chromosome 1 for this gamete. Should we take moms or dads? Doesn't matter. Okay, we need a chromosome 2. Moms or dads? Again, it doesn't matter. So let's bring that home with a probability problem, okay? If an organism has an N of 10, what's the probability that the sperm will have all 10 maternal chromosomes? Okay, N is 10, so if they're 2N, their 2N number is 20, their diploid number is 20. The probability that chromosome 1 is randomly chosen in meiosis, that goes into this sperm came from that individual's mom, 50%, right? Could be moms, could be dads, 50%. The probability that chromosome 2 is the one from mom, again, 50%. So the probability that getting chromosome 1 from mom and chromosome 2 from mom is 50% times 50%. This is what we call the product rule of probability, and I encourage you to Google that and watch a video or two about it. Basically, it means the probability of two different things both happening is the product of their individual probabilities, okay? The probability that I flip a coin and get heads, 50%. The probability that you then flip a coin and get heads, that was 50%. But what's the probability that we both flip heads? It's 50% for me, 50% for you. Multiply those, 25% probability that we get heads and heads, okay? So out of 10 chromosomes, what's the probability that all 10 in this sperm came from that individual's mother and not the father? Well, it's 0.5 times 0.5 times 0.5 times 0.5, right? 10 times. There's a 0.097% chance, or 1 in 1,000 chance when there are 10 chromosomes that they will all be from that individual's mother, okay? We've got 23 pairs to choose from. Think about how many unique possibilities there are, okay? All right, that's it for chapter 2.